Last up for discussion this week are eukaryotic cells. And you will remember that eukaryotic cells are cells which contain a true nucleus. That means that their DNA is contained within a membrane-bound structure. Okay, that membrane-bound structure is called the nucleus. And the other thing about eukaryotes is they also contain true organelles, which again you will remember are molecules that perform a certain function for a cell and which are contained within their own membrane inside the cell. The nice thing about having true organelles, the advantage of having organelles in eukaryotic cells is it allows for a great deal of compartmentalization. And that means that if I have a hydrolysis reaction occurring in one location inside the eukaryotic cell, I can still have a dehydration synthesis reaction occurring in another part of the cell, and they can be occurring simultaneously and not cancel each other out. So I can have incompatible reactions happening in separate locations, and it allows for me to have increased efficiency of reactions. We'll learn a little bit more about how we increase the efficiency of reactions when we get to talking about metabolism in a week or two. We think that eukaryotes, as I mentioned in the last part of this lecture, we think that eukaryotes are derived from a prokaryotic ancestor, most likely some sort of archaea cell, and an individual by the name of Margulis many years ago postulated that the way that this happened was through an event where we had an original prokaryotic cell, some sort of archaea type cell, and that cell had the plasma membrane fold in or invaginate and actually thus surround the nucleoid region and create a nucleus, and then it picked up another bacterial cell from the environment, and that bacterial cell probably had the ability to metabolize oxygen to make energy. And once that cell was taken into the larger cell, rather than being digested, it persisted inside the cell and became what we call mitochondria. It's possible too that another type of bacterial or prokaryotic cell that could photosynthesize was later taken into some of these cells and became what later is known as chloroplasts. So we think that the idea here is that smaller bacteria were taken or incorporated into a larger prokaryotic cell, and rather than being degraded, those tinier cells persisted inside the larger cell and became true organelles. Some good evidence that leads us to support this hypothesis is that both mitochondria and chloroplasts inside of our animal cells have their own separate DNA, and they have an ability to replicate, and we don't see that with other types of organelles. This hypothesis that we took in smaller cells into the larger cell and those smaller cells became organelles is called the endosymbiotic hypothesis. So what are these different types of organelles that are found inside of eukaryotes? Well, that's for you to discover. Rather than have me discuss all of the individual organelles and their functions inside of the cell, I will be this week dividing you up in class into expert groups where you will teach other members within your small expert group the different organelles. And I'll have more details for you about that in class. So for now, what I'd like for you to do is on your own read ahead about the different organelles that are found inside eukaryotic cells and be ready for your expert group assignment in class on Wednesday. Now, the last thing I want to talk very briefly about is something called the endomembrane system, which is found in all eukaryotic cells. And the endomembrane system is a system by which we involve a series of organelles in the process of manufacturing and exporting proteins from the cell. And the pieces and parts involved in the endomembrane system include the outer coating of the nucleus, which is called the nuclear envelope, right here. Something called the endoplasmic reticulum, which is a series of flattened membranes that you're going to learn about shortly. Another group of flattened membranes called the Golgi apparatus. And finally, a number of vesicles that allow for the export of proteins out of the cell once they are manufactured. Once you have a little better idea of the pieces and parts of the endomembrane system through your exploration of the organelles, you will be able to better understand what's called the secretory pathway hypothesis, which is basically a description of the events that occur that lead to the manufacture of protein and its eventual export out of the cell. But we'll get to that later. You need first to learn your different organelles. As I said, make sure you've read through the material on organelles before you come to class next week. And in class, I will be assigning you to expert groups and telling you what you need to do to prepare for your presentations the following week. I will see you then.